Great. Okay, so we're going to continue talking about membrane traffic today. We ended talking about coat proteins, beginning to talk about coat proteins, and how they form vesicles that bring proteins from one compartment to another one. We also talked a little bit about the kinds of evidence that cell biologists use to analyze all sorts of aspects in cell biology, and particularly coat proteins, GFP and immunofluorescence. So we're going to finish up talking about coat proteins, and then we're going to introduce a new important concept, which is how small GTPases are used to control the formation of a coat on a particular compartment. And this theme of GTPases you'll keep encountering in just about everything that we talk about in cell biology. So we'll talk about a specific small GTPase, but you'll encounter small GTPases and their principles throughout the entire course. We're going to introduce a new protein called snares, the proteins that control where a vesicle will fuse with the bilayer. And then we might get to post-translational modifications that happen in the ER, and maybe get to signals that are required for sorting, but that may be in Thursday's class. Okay, so this is sort of where we ended up. We were talking about clathrin, which was discovered by by Barbara Pierce at MRC, and then her postdoc made really nice antibodies that could stain clathrin and accessory proteins. So you may recall that clathrin is found both on the plasma membrane, and if you look at a cell like this, you can see dots all over the plasma membrane. Those each correspond to clathrin-coated pits. And then you can see sort of a vacant part, that's the nucleus. And next to each nucleus, you can see a concentration of clathrin that is adjacent to the nucleus. And what that represents is clathrin that's associated with the trans-Golgi network. So there's one clathrin, so clathrin's found both at the plasma membrane and at the trans-Golgi network. And there's one in general, most eukaryotic cells have one Golgi apparatus, and it tends to be next to the nucleus. So that's what you're looking at here. Here's the trans-Golgi network associated, here's the trans-Golgi network, here's the trans-Golgi network. Now those clathrin-coated vesicles are different from each other. The plasma membrane kind are responsible for endocytosis, the internalization of receptors from the plasma membrane. On the other hand, the ones at the trans-Golgi network are responsible, function in the secretory pathway, and they are responsible for bringing, we'll see later when we talk about lysosomal enzymes, for bringing lysosomal enzymes from the trans-Golgi network to the lysosomes. So how are these two clathrins different from each other? They're actually not different at the, next slide show this? Next slide is the next slide. Yeah, so here's the trans-Golgi network guys, clathrin, they're going this way. Here are the plasma membrane guys, they're going this way. And the way that these differ from each other is, you may recall from the end of the last lecture that clathrin is actually a two-layer coat. Clathrin is on the outer layer, and then there are small proteins called AP proteins that are in the trans-Golgi network and at the plasma membrane. And these trans-Golgi network ones are different from the plasma membrane ones. So Scotty Robinson, who was a postdoc in Barbara Pierce's lab, made antibodies and also GFP constructs later, and was curious about the difference between these two compartments. Could the clathrin guys how are the clathrin guys different from the trans-Golgi guys? And so her thought process was something like this: to do an experiment with the tools she had in hand. What experiment would best distinguish between the different clathrin-coated vesicles forming on the plasma membrane versus the Golgi? How are these different from each other? And I want you to look at these possibilities. We've been through all of these experimental tools, and decide what the best experiment would be to distinguish clathrin-coated vesicles on the plasma membrane versus trans-Golgi clathrin-coated vesicles forming at the Golgi. How are those different from each other? They go to different places, they're functionally different. So what sets them each apart from each other? And we're going to start with that Instapoll question. One, we definitely saw a shift to the correct answer. And I'm going to go through the back row, if you all are fine with that. So what experiment would best distinguish, because I already asked them what their answers were. What experiment would best distinguish between different clathrin-coated vesicles forming on the plasma membrane? One thing you could do is express GFP-tagged clathrin in cells, and then use fluorescent microscopy to look at clathrin localization in living cells. So is this experiment possible? Yeah, I showed you that experiment. And first person in the back row there. So you rejected that answer. So why didn't you think that was the best experiment? And you can say pass if you want.
Well, initially I went with C, but everyone else here, most of the people went with D. And we just thought it wasn't really going to show like a difference. They were the same, like proteins.
Excellent thinking, right. GFP clathrin would look just like this, except it would be moving around. It would be in living cells. Excellent thought. So you'd see ones on the plasma membrane. You'd see ones at the trans-Golgi network, but you wouldn't see the difference between them, which is the experiment we want. So an experiment might be possible, but may not be the best one to answer the question that you want. I like the way you're thinking. The other possibility is to clone genes for different adapters from cells, compare the gene sequences of different adapters. So these adapters would be the ones we talked about, AP1 and AP2 at the plasma. Maybe I didn't talk about those yet. Yeah, okay. So, so there would be, you could clone different, no wonder no one chose that one. So you could, there are different gene sequences that you could clone and you could compare the sequences, but that's not going to distinguish between the plasma membrane and the trans-Golgi network ones. The other thing to do would be a pulse chase experiment. You could label the proteins, definitely possible. All these experiments are possible actually. So you would feed the cells radioactive amino acid like S35 methionine. You'd chase it and then run a gel. But that wouldn't tell you, running a gel doesn't tell you localization. We're looking at what distinguishes the plasma membrane guys versus the other ones. And then the entire back row chose D, which is use antibodies for different adapters. Compare different antibodies with immunofluorescence microscopy. And what you're remembering perhaps from the last experiment was that clathrin is 2 layers. So there is clathrin as the outside shell, and then there are adapters that are in the inner shell. And if you raised antibodies against these different adapters, perhaps you'd see different localizations. And that's exactly what Scotty Robinson, the postdoc in Barbara Pierce's lab, did. So Barbara Pierce had purified clathrin-coated vesicles from pig brain, and then Scotty Robinson, the postdoc in her lab, who's now a really distinguished and famous scientist of her own, also in England, although she's American, but her lab's in England and she's lived there for 30 or 40 years now. 30 years probably. So she took those clathrin-coated vesicles and further purified what proteins were in them. And notice there were sort of 2 different kinds of these adapter proteins. And she raised monoclonal antibodies against them and then did immunofluorescence microscopy to ask where they were. So her hypothesis was perhaps these adapters are what distinct. She knew that the clathrin was the same because she had the image that showed that the image I just showed you. In fact, that's her picture. That's a slide she sent me. So that's clathrin on the plasma membrane, the trans-Golgi network. And then she raised antibodies against these adapters. She called one of them AP1 and the other one AP2 and said, is that what distinguishes clathrin from one clathrin from the other? And that is what the result was. So let me show you those results. The antibodies she raised against AP1 looked like this. So here you're seeing a cell right here. Here's another cell. This is probably 2 cells that are overlapping. Here's a nice cell right here. These are flat cells. And you can see that just the vesicles, well, you just see the sort of bright spots near the nucleus. So it looks like AP1 is the adapter that's associated with the trans-Golgi network. And the other adapter that she purified was AP2, and what she could see, we might need the lights down if we can control that. AP2 that she raised antibodies against were dots all over the cell, and you don't see a particular concentration around the nucleus. So that if you add up the discrete pattern of AP1 and AP2, what you see is exactly what she had before. And you could look at this with different antibodies if you raise them in different species. So you could see that the AP1 antibodies would co-localize with the clathrin dots on the plasma membrane and not, on the trans-Golgi network, and not the ones on the plasma membrane. And AP2 would be the ones here. So that's the actual experimental evidence from Scotty Robinson that showed how AP1 proteins are different from AP2 proteins, and also that's what distinguishes the two clathrin patterns. Questions?